Hi guys, it is week 9. It is embryology. It's the winter 2021. We talk about formation of the neural tube, neural crest cells. Very important lecture. These are board favorites right here, these neural crest cells. What do they become? And then we'll talk about just start talking a little bit about mesoderm differentiation. So where have we been? So we talked about how gene products in the notochord, the primitive streak, mainly the notochord, but primitive streak, AV cells, precordial plate, they're released and they soak into the ectoderm cells. And specifically some of those genes that do the soaking above are called noggin, chordin, and Cerberus 1. And they all turn off BMP. When you turn off BMP in a ectoderm cell, it goes back to its default state, which is that of neural tissue. And so this is how we formed the neural ectoderm cells. And we specifically made a neural plate. And here's a cartoon of anyone, you pick any one of those, noggin, chordin, Cerberus 1, diffusing out of the notochord and soaking into these ectoderm cells, and then they turn on genes within here that turn off genes, which morph them into a plate of neural tissue called the neuroectoderm. This is the brain and spinal cord right here, the very start of it. And then we talked about convergent extension, how the plate starts growing further and further caudally in this direction, but it grows tighter together, so you get a key formation. You don't get a wide formation like you do here in the future brain area. All right, so we made this last time. We talked about this neural plate, but it was flat and we didn't do anything. So now we need to fold it because, of course, the spinal cord is a tube, right? And the brain is kind of like a big tube in a way. So that's where we're going. We need to fold that plate into a tube-like structure. So let's talk about lateral folding in the neural plate. So about day 18, lateral folding is believed to start. And there's a bunch of mechanisms. I cut all this stuff out because it's just too complicated. They used to think it was a single mechanism that caused the folding, but it's not. It's a much more complicated story. And therefore we'll just say, based on numerous mechanisms, intrinsic and extrinsic, to the neuroectoderm cells, the darn thing starts folding. We'll talk about some specifics here in a second. But the very first thing to form is, or the very first thing to happen is the cells of the neural plate, which were cuboidal in shape, they undergo a transformation and become at the apex, they become very pointed, like a triangle shape or like this pizza here. And the neighbors, because of this, the neighbors are still kind of rectangular in shape. So that bends that flat neural plate into it starts to bend it in the middle. I'll show you a picture here of that in a second. So yeah, like a triangle shape, and it bends it downward. And here we go. This was the once flat neural plate. There's the notochord, there's mesoderm, endoderm. This is all ectoderm, but this is neuroectoderm because it transformed into the neural plate. And it was flat, and now it's not flat anymore because of this triangular shapeness that's happening right in the very midline, right above the notochord. And there is a gene product that you should know that sets this off. It's probably more complicated than this, but this is an easy one to remember. And it's called shroom. And it's an actin binding protein, and we'll leave it at that. The shroom soaks in. It's released by the notochord, cells in the notochord. The shroom gene turns on, and those notochordal shroom gene product goes up and soaks into the neural plate in the midline. And it causes apical narrowing to occur and that right in the very midline, and that's called the median hinge point, because now we have a kind of a triangular shaped neural plate instead of a flat neural plate. And the left and right sides of the triangle are called neural folds, and which create a groove in the middle, right in the midline, and that's called the neural groove. Let's look at a picture. So here was our fairly flat neural plate a little while ago, and shroom nodal cord was right here. Shroom soaked into these center cells and caused them to take on a triangular shape like these, and that bent the neural plate into a V-shaped. And these are called a neural fold, the right and left neural folds, and this is called the neural groove right here. And there's another thing helping this bend, and notice that it's bending downward into the mesoderm. The ectoderm is also growing. These ectoderm cells via mitosis are splitting and pushing in and pinching this and help driving it down. So that's another force that's going on. Ectoderm is helping. As the lateral plate is folding downward, ectoderm cells are also growing toward the midline, as I just explained, and it's kind of pushing that V downward. Okay. All right, so now we have a V, but that's not a tube, right? We need to make a tube-like structure. And so how do we do that? Well, we're going to do exactly the same thing. Hang on a sec here. I'm just got a sore back today, so I'm getting a pillow behind my back. There we go. That's better. Yeah, we're going to do the same thing in the lateral part of those neural folds. So right here, we're going to do the same thing, right? If we turn right in the center, if we turn these into triangular shape, right? What's that going to do? If we turn a couple of these into triangles, well, it's going to bend the tube like that, and so those are called lateral hinge points will form next, and it's exactly the same story. The shroom soaks up into those areas, and now we have more of a tube-like structure, and the neural crests or the ectoderm cells are still multiplying and pushing this way, so it's folding everything down. We also have some really incredibly important cells starting to transform here at the edge of these neural folds, and those are the neural crest cells, and those are incredibly amazing stem cell-like tissues. All right, so right as this lateral folding goes on, we get the neural crest cells, NCC, sometimes they're called. They're so important that some researchers call them the fourth germ layer, and we'll definitely come back to those. Okay, so let's finish up the tube and talk about these neural crest cells. So by day 21, this combination of hinge points and the ingrowth of the ectoderm pushing inward, we get a tube-like structure, and the neural folds are going to start touching each other, and they're going to fuse together. So we are going to create a tube-like structure. It's important to note that the whole tube doesn't close at once. More in the cranial region, this is where the first closing occurs, and there's a delayed closing of the rest of the tube. It kind of zips like a zipper going both ways. It zips to the cranial and caudal regions. So it happens more cranial region at first. But here we can see more hinges have formed, perhaps to help it form, and then we have our ectoderm growing. Bottom line is it's going to pinch it in the center, and it also sinks it as the ectoderm cells grow and grow and grow. They finally attach to each other, and now that whole section of the neural plate is gone, basically been swallowed into the mesoderm, because this is all mesoderm right here. All right. The other important thing is notice the neural crest cells here. Before this actually closes, they get the heck out of there. They become mesenchymal and have the ability to crawl. So right before closure, they migrate and they head right up above at 12 o'clock here of the newly created neural tube. They don't stay there very long. They really quickly, they split, and they'll go down kind of and hang out at this region in here with a little bit more time. Let's see. Yep, so everything I just said. Just before the tube closes, neural crest cells get the heck out of there. That has a name, the action of getting out of there, and that's called delamination. Delamination. That's a buzzword. Put that on a notochord. What is delamination? That's how the neural crest cells leave the neural plate tissue just before the neural plate folds into a neural tube or rolls up into a neural tube. That's called delamination. Got it? And there we are, about day 21, and I kind of looked at this picture all over. But the neural crest cells, how did they get out of there? Well, they had to, because these are epithelial cells, right? These are neuroectoderm cells, but they're epithelial in nature. So these cells went epithelial to mesenchymal transformation, which we've already talked about several times during the process of delamination. So the process of delamination is a epithelial to mesenchymal transformation process as well. And yep, that's the story of that. So again, as I said, the neural tube doesn't close all at once. It closes more in the cervical region first. So according to Langman, it's more in the cervical, which is more in the middle of the bilaminar disc at this point. And then it zips up to the caudal and cranial ends, and it leaves. I mean, if it's fused in the middle, if it's a tube-like in the middle, the open ends have names, and those are called the neural pores, where the neural plate hasn't rolled up into a tube yet. And the one toward the cranial region is called the cranial neural pore. The one toward the caudal region is called the caudal neural pore. Carlson calls these the posterior neural pore or the rostral neural pore. And that's just wrong more. That should be, rostral means toward the front of the brain, so, but that's how it is in Moore, so that's a book error. So they need to clean that up. And then the cranial neural pore sometimes is called the anterior. Remember I said anterior is a strange AKA for cranial in embryology. So it could be the anterior. If you're a medical student watching this, I'm pretty sure most of you guys use Carlson. So anyway, that's the story with that. And now we have a picture, as always best. So we have the closure, the tube rolled up right here. But you can see it doesn't happen all the way. This is actually the cervical region. This is all brain region up here, an occiput region. So it closes up first in the cervical region, and then it'll slowly close and zip as time goes by. But these open ends, these are called the neural pores. So the neural pore will get smaller and smaller and smaller, and you can see here the cranial neural pore is almost completely closed. Everything is zipped up. We'll talk about these somites next time. These are very, very important structures as well. It's paraxial mesoderm. We'll talk a little bit about how that's formed in a second. All right, I think that's all I needed to say about that. So some fun facts about the neural pores. I always ask this question: which one closes first? Because once neurulation isn't complete until the neural pores are closed. So who closes first? It's actually the cranial neural pore closes first. It's about day 25. The caudal neural pore closes on day 28. And once the caudal neural pore is closed, our process of neurulation is complete. So on day 28, 25 somite stage, and that you'll understand what I mean by that later. But that's a very important fact, is the caudal neural pore closes first, about day 28. And so now we have our central nervous system basically formed, and it's a tube. It's a hollow tube that has formed. And there's some major birth defects that occur if this tube doesn't occur. And I took a lot of this out. We'll talk maybe a little bit more in fifth quarter about this. But Gross slide, so be forewarned. So there's two words you got to make note cards and burn this into your brain. So if the cervical and kind of thoracic lumbar portion, or even the sacrococcyx, if they don't close, if the neural pore doesn't close and the tube never forms, that means that your vertebral, or your posterior arch, or your vertebral arch will never form, and therefore your spine will basically be open to your spinal cord, and that's a situation called a rachis. A rachis. You can say that 10 times fast. Okay. The same thing can happen up here in the brain. Now this is more the cranium. The bones of the cranium don't form, and your brain is exposed. That's called a cranioscus. Crane, like rain. Cranioscus. Cranioscus. Right? So, and we will definitely talk more about these rachis in, when will we talk about those? In 5th quarter. I don't know. We'll talk about them somewhere in my time with you. All right, let's look at these neural crest cells. Very important section of the class right here. The most important thing for board preparation is to know what these neural crest cells become, and know what mesoderm morphs into and ectoderm becomes an endoderm. Boards love simple questions because nobody understands this stuff. So these are really easy questions that professors can put together. So again, they're so important. They're sometimes called the 4th germ layer. These are mesenchymal cells, so they can travel, and they are super stem cells. They can morph into many different structures. Now, here's an easy question, or maybe not so easy. What neural crest cells were derived from what? What does that even mean, derived? Who was their parent tissue? What was the parent tissue that was derived? What was the parent tissue of a neural crest cell? Well, there's two ways. At the very basic level, they came from ectoderm. So neural crest cells were derived from ectoderm. If you want to get more specific, you could say, because where they really came from is neuroectoderm cells. So you could see either type of question depending how deep it is. Neural crest cells were derived from neuroectoderm or ectoderm. Recall what induces neural crest cells to form? Yeah, did we say this? I can't remember if we said this. But bone morphogenetic protein exposure is the key here. So ectoderm cells in the midline were exposed to BMP inhibitors, remember? No, I don't think we did go over this. We just said that to become neuroectoderm, you had to turn off BMP via exposure to choroid, noggin, and cerebrus 1. So the midline neuroectoderm cells don't secrete BMP, and they can become neural crest cells based on that. Or not, no, that's not true in 29. Because it's the ones in the tips. So I got to take that statement out there in 29. That's no bueno. Okay, but they don't secrete BMP, so they become neuroectoderm cells. We'll see right now how the neural crest cells. So what induces the neural ectoderm cells to morph into neural crest cells? Well, it turns out that the lateralmost regions between the ectoderm, the real ectoderm cells, which are secreting BMP quite strongly, and these neuroectoderm cells, which aren't secreting BMPs, these cells right on the fringe actually secrete just a little bit of BMP, or a medium amount of BMP, and that turns on genes within them. That medium expression of BMP is enough to turn on genes which we won't get into, but that's what wakes them up and makes them become neural crest cells, because of that expression, that medium expression. I don't think we'll get into that more. Oh yeah, maybe we will get into that a little bit more. The medium expression turns on the following genes. These are all transcription factor creating genes: MSX, DLX5, and PAX, PAX. And these factors go on to turn on even more genes, FOX3, SOX10, ETS. And this combination, you can see it's incredibly complex, actually creates the neural crest cell. I typically don't test on that. That's like really beyond the scope of, I think, primary healthcare providers. But there it is anyway. I don't even think a board would go that deep, but just in case, maybe a medical board might. So if you have one of those photograph memories and that's easy to memorize, well, that's great, but I think that's a little bit too much. And to make things even more complicated, Moore's, which is this chiropractic board book, says that Wnt and beta carotene signaling kind of wakes up or activates the GBX2 homobox gene. So this GBX homobox gene is also involved along with these other genes. So it's an incredibly complex story. The authors don't agree on it, so I think that's kind of beyond the scope. How do neural crest cells break loose? This is not beyond the scope, though. So we have to turn off the cadherins, the N-cadherins, maybe some E-cadherins as well, because they're attached to the the ectoderm cells as well. So basically, they get exposed to the CAM turner offers, SNAIL1, 2, TWIST, and FOXD3. FOXD3, that was actually already, just realized that was already included over there. So yeah, those are the ones that turn off, because we got to turn them. We're turning them into mesenchymal cells which can move, and they can't move if they're attached to the ectoderm or the neural ectoderm. They have to be free, and to free them, we need these genes turned on, and they need to release those gene products. Okay, great. So now they're mesenchymal cells. They've went epithelial to mesenchymal transformation. We've already said that, and they can migrate, and we said they do migrate. And remember, they migrate as the tube closed. I guess I could have put a picture in there, but we remember that picture. So they're out. They jump out of the tube right before it closed. There's some protectors of this migration process, because it's not easy migrating out of the neural tube and then migrating to your final destination. And so some of the cells migrate great distances, as we'll see. They have to migrate all the way down to your little toe, some of them, because nerves go down that far. So they have to be protected, and a SOC10 gene is the one that protects them. Was that on our list over here? It is on our list there. So that is also involved. So I guess we are knocking some of these out. So SOC10 protects them so they can make it to their predestined location. Mutations of the SOC gene will cause premature differentiation, and you'll just get messed up neural crest cells. So you need SOC10. See, I'm trying to pick all the genes that I can give you a nice visual to remember them by. All right, the guiding genes. So before the neural crest cells start moving, there are some guidance genes that have to be turned on that help them steer away from being destroyed. So, and this is too much here. Although FOXD3 is still there, SOC10 is still there, but RoboSlit, Neurofilin, and semaphorin and epherin are all these guidance genes, and they help the neural crest cells move to their location. I won't ask that question. But for those of you who have to know everything, what path do they choose? So where do they migrate? They don't migrate anywhere, because things like chondroitin sulfate, proteoglycans which contain chondroitin sulfate can destroy them. So they have to be careful where they go, and so they hug the surface of the ectoderm and the neural tube. So they migrate off, staying very close to the neural tube that kind of gave birth to them. And they can stay on the undersurface of the ectoderm as well. Specifically, there's a basal lamina that they migrate in, but I think that might be going too far. All right, big star here. You gotta guarantee you. I mean, if you prepping for boards, you don't have time, this is the stuff that you have to know. What do neural crest cells become? So amazing amount of tissue. I always think Well, they came from neuroectoderm, so it's going to be neural tissue, and that's pretty much true. Most of the neural tissue comes from these NCC cells, neural crest cells. So in general, they give rise to most of the peripheral nervous system and the autonomic nervous system. So your sciatic nerve, your obturator nerve, the cranial nerves, these are all built from neural crest cells, which get to an area and then they start multiplying and spreading out into a big nerve-like structure. But let's see exactly what they become. So ganglia, sensory ganglia, the dorsal root ganglia is derived from neural crest cells. The sensory ganglia of all the well, not all the cranial nerves, but 5, 7, 9, and 10, they're all derived from neural crest cells. Sympathetic ganglia, all derived from neural crest cells. The adrenal medulla is kind of a modified sympathetic nerve in a way, so it's no surprise the adrenal medulla, not the adrenal cortex though, but the adrenal medulla is derived from neural crest cells. The neural crest cells that hang out in the trunk region, not the ones from so much the cranial region, because neural crest cells will go the entire length of the neural tube. So the trunk neural crest cells, they give rise to your entire peripheral nervous system, you name it. Sciatic nerve, femoral nerve, obturator nerve, I just named the big ones. The radial nerve, the musculocutaneous nerve, the ulnar nerve, all neural crest cell derived. But there's some in the cranial region, they don't make all of the cranial nerves, the cranial nerves. There's cells called placental cells, which are stem cell-like as well. Or placode, sorry, not placenta. Placode cells. And they can also give rise to nerves in the cranial region. Now, I put this in here because this was kind of recently overturned, but Merkel cells, they were once thought to be derived from neural crest cells. In fact, Moore still has this, or is it Carlson? One of the big books still has this messed up because this has changed. Merkel cells no longer are believed to be derived from neural crest cells. It's all about the Merkel cells are derived from ectoderm cells, according to our chiropractic board book, Moore's, and they put forth good research. And I did look at the research, and it's true. Do you guys know what Merkel cells are? They're part of they're the epidermal portion of the Merkel disc. Remember the Merkel disc that gives you the ability to feel like it's in your fingertips? The Merkel cell, and then it connects to these A-beta afferent nerve endings. Let's take a look at a picture. So here's a Merkel disc. Here's the Merkel cell. This is in the epidermis, and this comes into the epidermis to connect with the Merkel cell. But these sense tactile stimulation. So this Merkel cell, was derived from neural crest cells. Okay, here's a picture of glabrous skin, thick skin here. How can I tell this is glabrous skin, by the way? Here's Dr. Doe lesson, probably. It's got stratum lucidum, right? Stratum lucidum is only found in the glabrous, or the thick skin, so I know this is thick skin. But here is a Merkel disc right here, and there's the nerve. So this little guy right here is the Merkel cell that was derived by neural crest cells. Schwann cells, which are super important. I'll talk about that when we talk about pernicious anemia. But they were also derived from neuroectoderm, or from, well, you could say they're derived from neuroectoderm as well. So you have to be careful on questions like that. What are Schwann cells derived from? The question could be really precise, or could be not precise at all. So ectoderm, yeah, that's true in a way. Ectoderm turns into neuroectoderm, but neuroectoderm turns into neural crest cells. So, I mean, all three of those typically can derive Schwann cells. See, we have to be very careful when you're forming a question or when you're answering questions about where things came from, because there's different levels. Schwann cells are amazing. They are like insulation around our nerves, nerves send electrical signals, and just like electrical wire, you better have insulation around them, or you're going to have short circuits and cause trouble. Multiple sclerosis deactivates these Schwann cells, and so you do get short circuits, and so does pernicious anemia. Neurolemma is the covering, so not only the kind of the meat of the Schwann cell, but the neurolemma itself. And I don't know if you've had this. You might not have. This is why this should be really in second quarter of this class, because I don't think you've even had this yet. But here's a run-of-the-mill peripheral nerve, like here. You pick one. Muscocutaneous nerve, how about? And then here is the insulation. There's a Schwann cell which wraps around and around. It secretes myelin, and it insulates these axons. And so these are derived from neural crest cells. And the kind of the membrane is also derived, called the neurolemma, is also derived from neural crest cells. Interestingly, though, all the other covers, like the endoneurium here, and then the perineurium, which surrounds a fascicle, which is a bundle of axons, and then even the epineurium, which surrounds bundles of these fascicles. Those were created by mesoderm. So watch out for that question. Autonomic nervous system, so sympathetic chain ganglia, you've seen those in anatomy. A celiac ganglia, you might have seen that in anatomy. Parasympathetic postganglionic neurons, parasympathetic ganglion, that you'll learn next quarter. The ciliary, ethmoid, sphenopalatine, submandibular. These are all made by neural crest cells. The skin is interesting. We'll talk about this more in seventh quarter. But the epidermis, which is the very top layer of skin, it has those 4 or 5 layers depending on what part of the body you're in. But that's derived from ectoderm. The mesoderm gives rise to the second layer down, which is the dermis. But that's not always true, so you have to be very careful if you're writing questions, because neural crest cells, they create the dermis of the face and the neck. So board UP people writing board questions have to be careful of that, because you can't say, dermis is created, all dermis of the body is created by what? Well, that's a trick question, because mesoderm usually creates dermis, but not in the face or not around the neck, and that comes from neural crest cells. The pigmented, so you have to watch out for this. Pigmented cells, the embryology authors say pigmented cells. That's not what you call these things. That's an old AKA. These are called melanocytes because they secrete melanin. So watch out for pigmented cells. It's a weird AKA used by some of these embryology authors. But they're also created by neural crest cells. These are really important, right? They look like little octopus. And their job is to, in the first layer, these are stratum basale has just given birth to these keratinocytes, and these neural crest cells put their little tentacles into the cells and inject melanin, and the melanin goes over the top of the nucleus to protect it from UV damage. And one of the greatest ironies in the world is that these melanocytes, they protect all these adjacent cells from UV damage by protecting the nucleus with melanin. Its own nucleus has no melanin around it. So go figure. I don't know why the committee did that. And that's what you've heard of malignant melanoma, is a cancer, a mutation of the genes in this melanocyte. It makes it go crazy, makes it become immortal, makes it divide too rapidly, and that's what a cancer is, immortal cells that divide too rapidly. Anyway, I'm getting out of control here. What else? What else do neuroectoderm cells create? Well, they create the leptomeninges of the prosencephalon, the forebrain, and the midbrain, the mesencephalon. What the heck are leptomeninges, you may be wondering. Leptomeninges is a way to say pia mater and arachnoid mater, not the dura mater, though. Okay, dura mater is out, pia and arachnoid mater. Got it? Let's see, what else do they make? The adrenal medulla, I think we said that, as well as the chromophene cells. which live in the adrenal medulla. The number one cell population in the adrenal medulla are chromaffin cells. Why do we care about them? They secrete something you've heard of, epinephrine. That's where epinephrine comes from, which has many functions. Speeds up the heart, shuts off the skin, makes someone who's fainting. You ever notice they get cold and clammy looking and they're still green looking? It's because epinephrine has shut off the skin, basically cutting off the blood supply to the skin to conserve that for fight or flight. Carotid bodies are also made from neural crest cells. Now here's where, because we've mainly been talking about neural tissue and cells, but here it also forms some bone. So the neural crest cells also form the calvarium, which is kind of the skull cap, if you will, the top of the skull. So the squamous portion of the parietal and frontal bones are both made from neural crest cells. The maxilla and mandible, these are next quarter. Yep, and I think this should be in second quarter, so don't get mad at me. But maxilla and mandible. The bones of the nasal cavity, ethmoid and lacrimal bones, all the little tiny bones you'll see. You've already seen these. The auditory ossicles, remember those? Where are those little things? The malleus, incus, and stapes, those are all derived from neural crest cells. And here's the skull. We'll actually do this, have a little bit of this in lab, which I kind of put in the last lecture. But there's the frontal bone, the squamous portion of the frontal bone, squamous portion of the parietal bone. Squamous is the major portion. There's a squamous portion, the temporal bone, and zygomatic bone. There's the mandible, there's the maxilla. Those are those structures. Ethmoid bone is there. Should be bigger. Artists made the lacrimal bone too big, but yep, so those are parts of the skull that are derived from neural crest cells. How do neural crest cells know to go, where to go? That's the $64,000 question. We don't know really what goes on. One theory an old theory has said that neural crest cells have equal potential to become anything, and they just blindly stumble into, let's say they blindly stumble into the place where the adrenal gland, or where the adrenal gland is, and they become the medulla. Because of the environment. It's chemicals in the environment that make them turn into what they do. So it's all about the biomechanical environment. Neural crest cells all have equal potential to become anything. Other theories say that's not true based on experimentation. You can't take neural crest cells that are destined to become the frontal bone. You can't put them in the adrenal medulla. They won't turn in to chromaffin cells. So there's research on that. So it's probably a mix of these two theories. The second theory says that they're pre-programmed to swim away from the neural tube and find their destination and become that tissue. So we just don't know the answer to that. Caudal extension. Now we've talked about this a little bit. We've talked about the regression of the primitive streak. So what's the deal with this neural tube? Let's go back to the neural tube because we have a little more work to do on it actually. So the sacral and coccygeal regions don't form during that first wave of neurulation. There's a second phase of neurulation called secondary neurulation that we've already talked about. So that involves the tail bud. Remember we talked about how the tail bud, the primitive streak, was dissolving backwards, but the last part of the tail bud out of the caudal region morphed into this weird kind of stem cell-like tissue called the tail bud. And so now cells from that tail bud tissue leave the tail bud, and they're mesenchymal cells. And we won't worry about the story, but they go down underneath and form this medullary cord right here. So here's the neural tube that we just built, but it's incomplete because the sacrum and the coccyx don't have one yet. So the tail bud formed here after the primitive streak disappeared, and cells swam out. You can draw some cells swimming out, and they went through epithelial to mesenchymal transformation, and they're forming a new tissue here. And that's called the medullary cord. The next thing that happens is it hollows out, and a kind of a central canal is created there, if you will. And then it connects to the real neural tube, and the two morph together. And that's pretty much the end of the story. So here's a cavitation is the process where this secondary neurulation, or this tube becomes tube-like, and then it binds with the real original tube, and you can see that they've joined here. And so now secondary neurulation is complete. What happens next? Well, we said that neurulation is over when all the neural pores close. The next thing that happens, once the neural pores close, the next big thing that happens is the cranial portion of the neurotube, which is bigger. Remember, the plate was bigger up in that region, so it's going to be a bigger part of the tube. That turns into a primitive brain. Specifically, 3 vesicles are formed. Now let's take a look at that. So we get a forebrain, the midbrain, and the hindbrain form out of that kind of wide part of the neurotube here. In the forebrain, you have to know this. This is absolutely mandatory. Prosencephalon is the forebrain. That's going to be frontal lobe eventually. And these are the embryologic brain that's forming. The midbrain, the mesencephalon, and then the hindbrain is the rhombencephalon. So that's the very first brain that's occurred, and we even have a little eye starting to form, which I'm not going to get into. And then after that, we're going to get some subdivisions occurring in the fore and hindbrain. So the forebrain, the prosencephalon, morphs into a diencephalon and telencephalon. Some say telencephalon, I say telencephalon. Why do we care about that? Because the diencephalon will later mature into the thalamus, the adult thalamus, or the live human thalamus and hypothalamus. Telencephalon will morph into the all-important cerebral cortexes. Okay, so those are super important. Mesencephalon stays the same. That's just going to become midbrain. I could have put that in there. The metencephalon, the rhombencephalon, morphs into a metencephalon and a myelencephalon. The metencephalon gives rise to the pons and cerebellum. And then the myelencephalon morphs into the medulla oblongata, which will morph into the spinal cord. It will connect with the spinal cord later. Got it? Memorize these slides. Note card slides right here. Make note cards. Right? So here's where we are now in a cross-sectional view or an axial view. We can see we have mesoderm. We have an ectoderm here. The neural plate is gone. The neural plate was here for a while, but remember we sunk the neural plate into the mesoderm and rolled it up into a tube, and then we covered the place where it used to be via mitosis of these ectoderm cells. So we have an ectoderm, we have a mesoderm, in the middle we have a neural tube, we still have a nodal cord beneath that, and then we have our endoderm down here, and this is our secondary yolk sac, and this is the amniotic cavity, and there'll be fluid in here, and that's where we are. So now let's do something with this mesoderm. This is the next thing, and there are other things going on at the same time, but about day 17, and remember the mesoderm, this is all mesenchymal. Now, it was once epithelial tissue, it morphed into mesenchymal tissue, and now it lives in between the ectoderm and endoderm, and it's mesenchymal. It can still move and change if it needs to, and that's we're going to use that here. So about day 17, the mesenchymal cells of the mesoderm, they start to clump into 3 regions. So we're going to form the paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm. So paraxial mesoderm will be right here. I think I have slides of this, but intermediate mesoderm, lateral plate mesoderm, which we're going to split into, we're going to kind of split this like this in a little while. But for right now, we have, on each side, we have this going on, right? So paraxial mesoderm, intermediate mesoderm, and this would be lateral plate mesoderm in a little while. Oh yeah, I don't know why I didn't go there. But there we go. All right, and now just a side note, we do have other events going on simultaneously. Gastrulation is done, right? Because we had to make mesoderm. We can't segment this mesoderm or clump these mesoderm into groups. We have to have it there first. gastrulation is the process of making the 3 germ layers, including the mesoderm. Primitive streak is regressing. The nodal cord is growing. Neurulation is just beginning. So we're kind of going back before the neurulation story here. Neuroplate is just starting to form. So what causes the mesoderm to differentiate? Oh gosh, here we go. It's another BMP, specifically BMP4 story here. And just like in the ectoderm, different regions of mesodermal cells have different amounts of BMP gene turned on, and they make different amounts of gene products, and that creates a gradient throughout the mesoderm. So and then the BMP will turn on different genes which create the different subpopulations. So let's look. So mesoderms near the midline secrete very low levels of BMP4, and that turns on certain genes and makes them become paraxial mesoderm cells, collectively called the paraxial mesoderm. The ones furthest away from the midline still secrete very high levels of BMP, and therefore it morphs them into the lateral plate mesoderm. And the mesoderm cells between the paraxial and the lateral mesoderm, they express medium amounts of BMP, and that turns on certain genes and morphs that mesoderm tissue into the intermediate mesoderm. Right? So there's kind of for you visual people out there, decreased BMP in the paraxial mesoderm. Kind of moderate amounts of BMP are expressed here. High levels of BMP are expressed out here. BMP4 here, and BMP is an amazing molecule. It's amazing in fusion, spinal fusion as well. People, smokers and people with osteoporosis who didn't used to be able to have a fusion can now have a fusion because of BMP. Got YouTube videos on that. That's way beyond you guys for right now. Paraxial mesoderm, what is it? What does it form? Where does it form? It forms everywhere, and this isn't true of all of these, but from the cranial, occipital, cervical, and the trunk. Another problem with the authors, the embryology authors, when they say trunk, they mean thoracic, lumbar, sacral, and coccygeal area. I don't know why they do that, but even Carlson does that. So watch out when they're talking about the trunk. They kind of lump the thoracic, lumbar, sacral, and coccyx all into one area. So paraxial mesoderm forms everywhere. There it is again. There's the paraxial mesoderm. Why don't they secrete BMP4? We said that BMP4 is not secreted very low levels in these mesoderm cells that become paraxial mesoderm. Because cells near the midline were exposed to high levels of noggin, chordin, and cerebrus 1. And recall that these gene products, which are transcription factors, they block the attempts to turn on the expression of BMP4. So mitogenesis. So after the paraxial mesoderm forms, and this is what we're going to talk more about next week, after the paraxial mesoderm forms, it quickly clumps into blocks. So in order to clump, cams have to develop, and it has to go back into epithelial. So it goes through a mesodermal or a mesenthelial to epidermal transformation, and they get clumping. And we can see these, even with a powerful microscope, we can see these clumping, and those are called somites or somitomeres, depending on which region. We'll get into them next. What's the big deal about the intermediate mesoderm? What's the take-home message? What does that become, by the way? It's the urogenital system is what that becomes, but I'm kind of jumping around here. So about day 17, just to the outside of the paraxial mesoderm where medium amounts of BMP, you get the intermediate mesoderm. And this only forms in the cervical and trunk region. It doesn't form in the cranial region at all, which was not true for paraxial mesoderm or lateral plate mesoderm. Both of them form everywhere. But for whatever reason, we don't need intermediate mesoderm. There's only two divisions up in the cranial region. No intermediate mesoderm in the cranial region. Got that? So it segments as well, but it only confusingly, it doesn't segment everywhere like paraxial mesoderm does. It only segments in the cervical and upper thoracic regions. And it's non-segmented in the trunk or the lower thoracic, lumbar, sacral, and coccygeal regions. There it is, intermediate mesoderm. And what does it become? I think kidneys. The urogenital system is what the intermediate mesoderm ultimately morphs into. So in the cervical region, the cells there, they form the primordial kidney, all the parts of the kidney. In the lateral cervical region, the cells differentiate into something called the pronephretic duct, and that's the organizer. That organizes the entire urogenital system, and we'll just leave it at that. You're going to have to do some self-study on this because there's no way I have time to get into all this stuff. All right, so that's it. We're going to talk about next week some really important stuff, and that'll actually be our last lecture. I actually have, I think, 12 lectures. There's other lectures on YouTube. Just not enough time to get everything done in this class. All right, see y'all later.